All right, everyone. What I'm going to do is I'm going to break your Unit 3 exam review up into two groups. We'll do Chapter 7, which is going to look at the cellular respiration. And then in the next video, we'll look at mitosis and meiosis together. So if we're going to spend some time here in Chapter 7, we want to first look at the chemical reaction for respiration. So here on the screen, you can see that it is sugar plus oxygen, and this sugar is glucose. That's what your body prefers. We're going to take both of those in, and then we're going to create carbon dioxide, water, and release energy. So this is the chemical equation for respiration. Just like you saw in the last exam where you had to pick it out for photosynthesis, you're going to have to do the same thing here. Now, again, like the last exam, it's important to know what oxidation and reduction means. So if we look here, the sugar is going to be oxidized. It's going through oxidation because it's giving up the hydrogens. So who's donating the hydrogens in respiration? It's going to be glucose. That's important to know, that glucose is the one donating or providing the hydrogens. On the other hand, oxygen is going to be accepting those hydrogens, so this one is actually being reduced. So oxygen to water is going to be reduction. Okay, so this is reviewing those two terms, oxidation and reduction, as well as how it applies to respiration. Let's take a closer look at the steps of cellular respiration. So if you look here with cellular respiration, we've got the main steps that are located here. You have glycolysis. Then you're going to see the prep stage, which is not quite labeled here because it does not produce ATP, the Krebs cycle, and then the electron transport chain. So if we take a look at this, this is looking at the amount of ATP made in each step. The first one here is glucose. Glucose is going to produce 2 ATP, and it's going to do it through substrate level phosphorylation. Now remember, this substrate level phosphorylation means it's going to use enzymes to help with that, but oxygen's not present. We're not going to use chemiosmosis or anything like that. It's just an enzyme helping create that ATP. On the other hand, for the Krebs cycle, we also create 2 ATP, again at substrate level phosphorylation. The last stage here with the ETC, you'll notice that it actually produces a lot more ATP, 32 to 34, but it does it through oxidative phosphorylation. Now in this case, that's where that transfer of hydrogens is going to come in, where we have those electron carriers of NADH and FADH2. They're going to be sending those hydrogens over to do chemiosmosis to create a maximum amount of energy here, 32 to 34 ATPs. So guys, it's important to know which ones do substrate level phosphorylation and which ones do oxidative phosphorylation. Now, keeping that same picture here, we're going to put here these steps: what goes in, what comes out, where it occurs, amount of ATP, but also if oxygen is required. So let's take a look here with glycolysis. Guys, with glycolysis, glucose goes in and pyruvate is going to come out. Glycolysis actually means the splitting of glucose. So we're going to break it in half to create pyruvate. This happens in the cytoplasm of the cell, so make sure you know that that's where it occurs. And there is no oxygen required here. Okay, glycolysis is actually a form of anaerobic respiration where oxygen is not needed. Fermentation would be another example of anaerobic, no oxygen needed. Now in this case, we're going to make 2 ATP out of this step. Now the pyruvate that was created in the glycolysis is going to then be sent into the prep stage. At the prep stage, we're going to take pyruvate and we're going to change it a little bit, add some stuff to it, and it's going to be converted into acetyl-CoA. This is going to take place inside of the mitochondria, and more specifically the mitochondrial matrix. That's the fluid inside the mitochondria. Now, this is going to be where oxygen is required. Oxygen's like the ticket to get into the mitochondria, so it is required, but it is not used here. There's no ATP made in the prep stage. The next stage is the Krebs cycle. We're going to see that acetyl-CoA is going to be added, okay, to another molecule, which is going to be this oxaloacetate, and we're going to see that it's going to go through the Krebs cycle and create citrate. Now this is going to happen again in the mitochondrial matrix. Oxygen is required but not used, and we're going to make 2 ATP. The last step here is the ETC, the electron transport chain. This is going to take hydrogens from each step that were carried by NADH and FADH2. They're going to drop them off and they're going to be added to oxygen. When they're added to the oxygen, it's going to be converted into water. This is going to happen in the mitochondria cristae. This is the folds in the mitochondria. Oxygen's required, and this is where oxygen is going to be used. This is going to make 32 to 34 ATP, the largest amount. Okay, so it's important to know where each of these takes place, whether oxygen's required or not, and how many ATP come out of it. There's going to be several questions that deal with each of those. Now let's take a closer look at the inputs and outputs for each step, because we'll need to know those as well. So guys, here the first step is glycolysis. This comes straight out of your notes. Glycolysis has glucose going in, pyruvate coming out. But guys, I told you in class that it's that whole concept of it takes energy to make energy. So ATP is also going to go in, as well as 2 are going to be gained coming out. And we also have 2 NADHs. Remember, NADH is going to be that hydrogen carrier, that taxi cab that's going to take those hydrogens to the electron transport chain. The next step is the prep stage. The prep stage takes place in the mitochondria, so you can see that it's drawn here in the picture. Pyruvate is going to go in. What's going to come out is acetyl-CoA. Now there's 2 of them that are doing this, because remember, glucose got split in half. We also see there's carbon dioxide that comes out. This is the first step. You see the byproduct of carbon dioxide. There'll be one other one, but this one is where carbon dioxide comes out first. We also see that NADH is going to come out of here as well, going to transport those hydrogens to the electron transport chain. Now this brings us to the Krebs cycle. The Krebs cycle is going to take that acetyl-CoA and it's going to add it to oxaloacetate. This is going to convert it to what we call citrate. It's a type of citric acid. As it goes around this cycle, which you don't have to know all the steps, we're going to see that NADH comes out, FADH2, again, both hydrogen carriers, ATP, we're going to have 2 ATP, and carbon dioxide. So carbon dioxide, guys, is produced in the prep stage and the Krebs cycle. It's also important to note for each glucose, the Krebs cycle is going to turn twice. The last step is the main step that's going to create that large amount of ATP. Now this is going to be a set of proteins that are through that membrane of the mitochondria, which are going to move hydrogens and electrons. This is that chemiosmosis. This is going to allow for oxidative phosphorylation. So NADH and FADH2 are going to drop off their hydrogens like you see here. Okay, they are then going to go through the electron transport chain. As they do this, they are going to move these hydrogens to one side where we're going to force them into a high concentration. They're going to then want to go through this enzyme here, which is ATP synthase. As they do this, ATP synthase, of course, is going to make ATP, 32 to 34 ATP. Some of these hydrogens are going to then be accepted by oxygen. Oxygen's going to go in. When they accept those hydrogens, we're going to have water come out. So it's important to look at what goes in and what goes out. So that sheet, guys, that you guys filled out that looks like this, that has all the inputs and outputs, it's one single sheet. This is going to be a really good study tool for this exam. Now, this next slide, guys, is looking at the benefits of fermentation. Now, fermentation, guys, is an anaerobic process, meaning there's no oxygen that's going to be there. Aerobic, like we saw before, is oxygen is going to be present. Okay, but we want to look at the advantages and disadvantages of fermentation, because there's times that oxygen might not be present. So when we look at this, an advantage is that it gives a rapid burst of energy. Okay, it can give us energy quickly, even if oxygen's not present. The problem, though, the disadvantages is it's short-lived. It does not last very long. Cells will start to die and things like that if oxygen is not present. It gives low ATP. It only makes 2 ATP instead of what we saw there with that 36 to 38 ATP total when you added them together. It also is only 2% efficient. It's not very efficient. We saw that cellular respiration with glycolysis, prep stage, Krebs cycle, and ETC, that's going to be about 36 to 38% efficient. 2% not so good. It's also going to produce some toxic byproducts. Okay, for our cells, that's what we call lactic acid. It can build up in there and it can be toxic to our cells. Now, there are also some other benefits or advantages to fermentation when we talk about it economically, using it for making money. A lot of times we use it for food, things like beer, wine, and bread. We also use it to make cheese, yogurt, sauerkraut, soy sauce. The list can go on. It's also used a lot of times for types of cleaning supplies like acetic acid and that sort of thing. So there's some advantages and there's some disadvantages. Okay, there's a short answer question about this where you're going to be asked to discuss one or the other, or maybe both. Give me advantages and disadvantages. So take a look at these for fermentation. The last slide that I have here, guys, is that you need to make sure that you can identify the parts of the mitochondria. Now, there's two main structures you need to look at, and this is the cristae and the matrix. The cristae, guys, is the folds of the mitochondria, whereas the matrix is the fluid. Okay, so make sure that you can label these two structures of the mitochondria. Okay, so this briefly discusses what you need to know over chapter 7, which is cellular respiration. I know that it's taking it down to kind of a more simple level, and I talked about it quickly, but like I said, that handout that goes over the inputs and outputs, that's going to be very beneficial. Not only are you going to have some multiple choice over this, you will have some matching, like matching up the number of ATP for each step, matching the stage to what's happening, or maybe where it's located. We talked about location, cytoplasm versus the mitochondria. So make sure that you take a look at each of those types of areas. If you have any other questions, please let me know.